Good morning, my dear students. We are entering into the arena of pharmacodynamics. Pharmacodynamics means what drug does to the body. It includes the study of biological and therapeutic effects of drug, including their metabolism of action in the living system, relationship between dose and effect of the drug, and modification of drug effects by other drugs, and various environmental and patient related factors. A drug cannot create any new function in the body, it can only modify the existing pathological state for the benefit of the recipient. There are two terms which are existing in this regard. One is called affinity and another one is called intrinsic efficacy. By the term affinity, we mean the ability of a drug to bind with its receptor. So now we heard a new term, receptor, and by the term intrinsic efficacy, we mean the ability of the drug to bring some kind of subsequent changes in the tissues as a consequence of drug receptor interaction or drug cell interaction, and therefore will get different action. The actions are usually of two types: primary action, which is the action produced by the drug in unaltered form, and the secondary action, and that is the action produced by the drug in altered form. So the drug may produce the effect by stimulation, depression, irritation, counter irritation of specific tissue, replacement, and anti-infective effects and modification of immune status. Now, when we are talking about these pharmacodynamics, we also have to know what is a receptor. So, a receptor is basically a macromolecule and a protein. If you want to give it a definition, you can define it in various ways. The best possible way is probably to define it as a macromolecule or binding site located on the surface or inside the effector cell that serves to recognize the signal molecule or drug and initiate the response to it, but itself it has no other function. Though in broad sense all types of large biomolecules, including the effectors, enzymes, channels, transporters, with which a drug can bind to produce its action, have been denoted as receptors. Such designation tends to steal the specific meaning of this important term. If supplied, xanthine oxidase would be the receptor for allopurinol. L-type calcium channel would be the receptor for nifedipine. Serotonin transporter, that is SERT, would be the receptor for fluoxetine. But it is therefore better to reserve. The problem is the connotation is not in consonance with the general understanding of the term receptor. So therefore it is better to reserve the term receptor for purely regulatory macromolecules which combine with and mediate the action of signal molecule, including drugs. Physiologically, we can classify drug receptor into following types: the regulatory receptor, the enzymatic receptor like tyrosine kinase, which is the receptor for insulin, the transport receptor like sodium potassium ATPase, which is present in the cardiac cell, the structural receptor. So these are from cell parts, but these are the classification based on, as I said, on the basis of physiological system. Now, when we know about the receptor, we must also know how these different types of receptors are actually classified on the basis of signal transduction. On the basis of signal transduction, it can be classified into four types: the G protein coupled receptors, the ion channel receptors, the enzyme linked receptors, and the nuclear gene, that means intracellular receptors. Now, the G protein linked receptor means the single polypeptide chain threaded back and forth, resulting in 7 transmembrane helix. There is a G protein attached to the cytoplasmic side of the membrane and functions as a switch. So, this is the way these G protein channel receptors act. Here you see at the right hand side, the chemical signal binds as a ligand to the G protein linked receptor, and then the G protein linked receptor changes its shape and interacts with the G protein. Interaction causes a change of GDP to GTP. It is displaced and the GTP to be bound to the G protein. The active G protein now binds to another protein, usually in enzymes. From there, the enzyme is activated and ultimately the G protein is hydrolyzed. GTP is back to GDP and the G protein releases from the enzyme and the reaction stops. So that is the function of G protein coupled receptors. Most of the receptors like muscarinic, nicotinic, that means the cholinergic receptors, the beta alpha beta receptors, the histamine receptors, most of the receptors, benzodiazepine receptors, most of them are G protein coupled receptors. Second group of receptors are called the enzyme receptors. One such example is tyrosine kinase receptor. Structurally, they exist as individual polypeptides. Each has an extracellular signal binding site and intracellular tail with a number of tyrosines and a single helix spanning the membrane. Here you will see this is the extracellular site where the signal will come and bind. There is a gap between the two. So the ligands bind to both the receptors. The two receptors polypeptide aggregate. They will aggregate and form a dimer. So a dimer is formed and it's followed by the activation of tyrosine kinase in different parts of this dimer, and this will ultimately lead to phosphorylation, cellular response using ATP. Usually it's happened the tyrosines on the tail of the other polypeptide. So receptor proteins are now recognized by relay proteins inside the cell. Relay proteins bind to the phosphorylated tyrosines and may activate 10 or more different transduction pathways. So this is a function of enzymatic receptor. The first one was G protein coupled receptors. This one is enzymatic receptor, and the example is tyrosine kinase, which is a specific receptor for insulin. Next comes the ion channel receptor. There are a few receptors like NN receptor, that is nicotinic neuronal receptor. There is a receptor like GABA A receptor, which is a receptor for benzodiazepine, and there are certain receptors like glycine receptors, where we'll see the signal molecule binds as a ligand to the specific site of the receptor. What happens? The channel opens, conformation will change, and therefore there is a opening of the channel and the ion is coming inside the cell. If it is a negative ion coming inside, it's called hyperpolarization. If it's a positive ion coming inside, it may cause depolarization. So the change in ion concentration within the cell triggers the cellular response. So this is the third type of receptor as per signal transduction, and that is ion channel receptor. The last type of receptor is called the intracellular receptor. They are present usually in the plasma membrane. Same are the proteins located in the cytoplasm or nucleus of the target cell. There are different receptors like the steroid receptors, progesterone, testosterone, estradiol, thyroid hormones. So, all these are examples of intracellular receptors. Normally, many of them for their action required second messengers. So, second messengers are small non-protein water soluble molecules or ions readily spread throughout the cell by diffusion. Two or more widely used second messengers are cyclic AMP and calcium ions. Beside this, there is another one which is very commonly used as second messenger, that is inositol diphosphate and diacylglycerol. Calcium more widely used than cyclic AMP, used in neurotransmitters, growth factors, and some hormones. Increases in calcium causes many possible responses and muscle cell contraction, secretion of certain substances, and cell division. So, there are two benefits of a signal transduction pathway. The first one is signal amplification, and the second one is signal specificity. Amplification means proteins persist in active form long enough to process numerous molecules of substrate. Each catalytic step activates more products than in the preceding step. So, this is one example or the pictorial presentation of how from one molecule we are getting so many number of molecules, and this is happening via signal transduction pathway. So, on the basis of the fact whether a drug is having affinity and intrinsic efficacy or both, it can be classified into following types. The first one is called agonist, rather a pure agonist. What is that? This is an agent which activates a receptor to produce an effect similar to that of the physiological signal molecule. And here affinity is +1 as well as intrinsic efficacy is also +1. By the term affinity, we mean the ability of a drug to bind with its receptor. If you compare it with a plug point and a plug, you see the plug point is the receptor and plug is basically the drug. So, when the plug is joined with the plug point and you put on the switch, this you may see that your home is basically full of light. So, that indicates that this plug here is acting as an agonist, because it has both affinity as +1 as well as intrinsic efficacy as +1. Antagonist, by the term antagonist we mean an agent which prevents the action of an agonist on a receptor or a subsequent response, but does not have any effect of its own. It means the drug has full affinity, that means it is capable of binding with the receptor, but no intrinsic efficacy. Hence, the drug, again I am comparing, I am giving the same example. The plug is added with the plug point. Now you put on the switch. The light is not on. So, from there you can understand that in spite of binding of the plug point with the plug, the activity is not served. So, that is the role of antagonists. It is basically blocking the role of agonist. Partial agonist. This is basically an agent which activates a receptor to produce submaximal effect, but antagonizes the action of a full agonist. So, it can serve two purposes. It has partial antagonistic action as well as a partial agonistic action. And the last one is called inverse agonist. Here, an agent which activates a receptor to produce an effect will go in the opposite direction to that of the agonist. What does it mean? Its affinity is +1, so binding ability is +1, but intrinsic efficacy is going to be -1. It's going absolutely in the reverse direction of the agonist. Whereas in case of partial agonist, you will see its affinity as well as intrinsic efficacy will be between 0 to +1. And what is ligand? Any molecule which attaches selectively to particular receptor or sites. The term will indicate affinity or ability to bind without regard to functional change. An agonist and competitive antagonist are both ligands of the same receptor. So, in my next class, I will come up with this the concept of receptor occupation theory. But for the moment, you just try to understand that the agonists are of two types. The full agonist is having a maximum efficacy. Partial agonist obviously have a partial efficacy. So when you plot a log dose response curve, you will see that the difference. The difference is the efficacy or the maxima achieved by this full agonist is more in comparison to the partial agonist. Antagonists interact with the receptor but do not change the receptor. They have affinity but no efficacy. Usually they are of two types. The pharmacological antagonists. There are different types of antagonists that we will discuss again later. Pharmacological antagonists usually are of two types, competitive and non-competitive. The competitive antagonists, they compete with agonists for receptor. They are surmountable with increasing agonist concentration, displaces agonist dose response curve to the right, so it's a right-sided parallel shifting of the curve, reduces the apparent affinity of the agonist. Whereas a non-competitive antagonist binds to the receptor, stays bound, irreversible, does not let go of the receptor, produces slight dextral shift in the agonist, dear curve in the low concentration range. This looks like competitive antagonist, but as more and more receptors are bound, the agonist drug becomes incapable of eliciting a maximal effect. So the curve is not being shown here. The curve is not shown here, but I can tell you that from this curve, this efficacy level will come downwards in case of non-competitive antagonist, whereas in case of competitive antagonist, it will shift towards right hand side. So in case of competitive antagonist, you will see there is a right sided parallel shifting of the curve, and the maximum effect remains same when you are increasing the concentration of agonist in presence of antagonist. Even if you increase the concentration of antagonist further and also the agonist, it will be shifted towards right hand side. If the curve is shifted towards left hand side, it is called synergism. Again, that topic will be discussed later. If this curve is straight away coming downwards and touching the baseline, in spite of increasing the concentration of agonist, you can call it non-competitive antagonism. So for you at the moment, the pharmacological antagonism are of two types, competitive antagonism and non-competitive antagonism. So with this, I am concluding my first presentation on pharmacodynamics.